Hello, good day to all. This is Meraj before you, and we are starting with human reproduction today. And before this, I would request you all please go through my previous videos and especially the video on the concept of reproduction, so that you can understand better before this. So, with this, let us proceed with our class. So, my dear friends, when we come to the start the human reproduction study, so we come across the basic question arises is that what type of reproduction? What type of reproduction is this? Is it sexual type or asexual type? So the answer to this can be understood by knowing that we name sexual when there are two organisms taking part, and asexual when only one organism is taking participation during reproduction. But as we know that in the human's reproduction, intercourse is a stage that is very important for reproduction, in which the male transports its male gametes, that is the sperms, into the female vagina. By this intercourse, it means that there is a participation of two organisms. Two organisms are participating. That is the reason why we named that it as a sexual reproduction. So after understanding this, we will proceed to learn what are the reproductive events. So so first, it is the insemination. Insemination, the transfer of semen from the male to the female, and this is accomplished by the process of copulation, that is the intercourse. And after the insemination, after the male gamete is transferred to the female, and as it reaches to the fallopian tube, where the ovum is also present, that is the female gamete which is coming from the ovary. So here the fusion takes place. That process called the fertilization. And as we all know, the result of fertilization is the formation of zygote, that is diploid, because before this the gametes are haploid. So after fusing both the gametes, male and female, the zygote is formed, that is diploid. And this diploid zygote will undergo mitosis and go through stages of development, and the most important is the blastocyst stage, because this is the stage in which it gets implanted into the uterine wall. That is the process is next is the implantation process. And after the implantation, the development of the embryo takes place into the fetus and so on. So that period is called the period of gestation, that lasts nearly 40 weeks or 9 months. So in this gestation period, the development of embryo takes place into the fetus, and as it goes at the end of the gestation period, the parturition occurs. That is the parturition parturition occurs parturition occurs, and that is the birth of the newborn occurs. So this is the reproductive events, and these are very important to understand before going in depth about and learning about the fertilization and the implantation and the development of the zygote into the blastocyst and the process of insemination and fertility disorders and those all things. So basic knowledge we have to have basic knowledge, that is these are all the reproductive events. And now the question arises that from where are these gametes or the sperms being produced? So the answer to that, generally gametes might be and ovum, sperm and ovum are being produced. The answer to that question is that they are formed from the gonads. Gonads are the reproductive organs. They are present both the sexes. In males, the gonads is named as testis, and in females that is the ovary. So these are the reproductive organs. They are producing the gametes, and the process that process known as the gametogenesis, which we will be discussing in in our next videos in detail. At the same time, in next videos of embryology, all the process of implantation and the zygote development and the fertilization all will be discussed in detail. Further, you have to watch next videos. And about the testis, we have made a separate video of anatomy or its blood supply and clinical knowledge, venous supply, that is very most important differentiating feature and characteristic feature of the testis, and all those things have been discussed. You can go and watch varicocele and hydrocele and those all things. So these are all the things. And some one extra point you have I want to add is that if you take the semen, this is the insemination, means process of transfer of the semen from the male to the, and this what does if you take the one milliliter of the semen, you will and count all the sperms in them, so you will get nearly 15 to 200 million sperms. So in normal male individual, the sperm count is nearly 15 million to 200 million per ml of the semen. So that is a extra point. So with this, let us move next and study in detail about next things. So now even in secondary school we know that there are what are the types of organs. Here the organs means a reproductive organ. So generally we know that there are two types. One is the primary sexual organ and another is the secondary sexual organ. So in primary sexual organ, what is the difference between primary and secondary sexual organs? The many students get confused, but there can be no confusion remain after you get the real concept behind this primary and secondary sexual organ. And here primary sexual organ means they are primarily important because they are involving in the formation of the gametes. Gametes are very important, primarily important. So that is the reason why these are involved in the formation of gametes. So these are only gamete producing organs. These are only gamete producing organs. And whereas the secondary sexual organs, they are which differentiate, these will differentiate male from female. So they are involving in sexual, leading to sexual dimorphism. So my dear friends, the basic concept we have understood that the primary sexual organs are only gamete producing organs. So what are the gamete producing organs? Just now we have learned that is in male that is testis and females it is the ovary. And what are the secondary sexual organs which are differentiating male and female? They are the external genitalia. So they are named accordingly that if they are primarily important, they are known as primary sexual organs, and then the secondary sexual organ. So with this, let us move forward and try to learn what are the components of male reproductive system. So male reproductive system is also known as the male genital system. And what are the components of this system? The components are first are the sexual organs, and the second is the accessory might be accessory ducts or the accessory glands, and the third is the external genitalia. So all these are for the components of the male reproductive system. So in male reproductive system, the sexual organs, as we know that the sexual organ is in male is generally testes. In male it is the testis and female or the ovary. But here what is the site of this organ? Generally it is in the pelvis. But in case of male, these testes is descended and descending into the outside, descending into the scrotum and coming outside the body. The site is changed and now it is in the scrotum. Now let us know what are the accessory ducts. We all know that the accessory ducts are starting from epididymis or the ductus deferens, and then the ejaculatory duct, and then the Yes, all these are come down, and the so all these come under ducts. And what are the glands? We all know that glands can be prostatic gland, to name few, the seminal, vesicle, and some one another gland is also added, that is the bulbourethral gland, whose secretion is very negligible in the semen. So all these are the accessory. Now what is the second? What are the external genitalia in case of males? That is the penis and scrotum. So these are all the components of reproductive system. Now after going to the basic structure, now we will be focusing on the accessory glands in detail, and ducts and the glands in detail. Whereas the about the testes, we have already taken a class of anatomy on all. We have discussed all structures and the functions and as well. So please go through that. So with this, let us move forward. So now, before going to learn about the accessory glands and their secretions, now we have to focus on one important thing, that is the passage of spermatozoa. As we all know that the spermatozoa is formed in the seminiferous tubule, but in seminiferous tubules by a process of spermatogenesis, which is discussed in another class. Please go and watch there. And from so to the point, from the seminiferous tubules, the spermatozoa is coming out, and here it is passing to the reti testes, and from the reti testes it is moving to the ductus efferentia. This is the ductus efferentia. From here it is going into the epididymis, and from epididymis it will go into the ductus deferens. This is the ductus efferentia. This is the ductus deferens. So from the ductus deferens it will go into the ejaculatory duct, and from there into the urethra, and at last come out from the urethral meter, that is the external opening of the urethra, that are not mentioned indicated here. So this is the passage of spermatozoa. But to be specific, I will write here. I will explain you here. You please focus here. I'll be explaining in detail. So let us move. So from, as we all know, seminiferous tubule. The spermatozoa comes from the seminiferous tubule by the process of spermatogenesis from the spermatogonium, that is from the germinal epithelium. For details, you watch another video. From here it will come into the tubuli recti. To be specific, we have to write tubuli recti, then the rete testis, and after rete testis it will go into the, as we all know, vasa efferentia. And all these three constitute what? They constitute, as they all three are present inside the testes, this is called intra-testicular, genital duct system. These are come under the intratesticular genital duct system. After vasa differentia, let us move forward. After this vasa differentia, we know that it moves into the epididymis. But to be specific, we have to write that it is first caput, then corpus, then cauda, epididymis. All these are the parts of the epididymis. You have to be very specific, so we have to write all this. And then it will move to the vasa deferens, as we all know. And after vasa deferens, it will join with the duct of seminal vesicle to form ejaculatory duct. And up to ejaculatory ducts, every duct is paired, because there is right and left. But after the ejaculatory duct, both the sides unite to form the urethra. And for your knowledge, urethra can be studied under three. First is shorter proximal prostatic urethra. First part. Then it is very short middle membranous urethra, and last long penile urethra, and after this it will come through the urethral meters, external urethral meters to outside the body. This is all the passage of the spermatozoa that is going from the seminiferous tubule to the external body, and if the copulation occurs, it will enter into the vagina of female vagina. So this is all the passage of the spermatozoa. One to add a few things, I would like to discuss what are the function of the epididymis. The most two important functions to name are: epididymis provides space. It provides the space, storage space for sperms, so that it get time to mature. So these are the important points about the epididymis you have to remember compulsorily. So up to here there will be right and left parts. So right ejaculated, left right faucet different, left faucet different, right efferentia. So all these will at the point after the ejaculated duct, they will get united to form the urethra, and urethra can be divided into parts, that is the prostatic, shorter prostatic, then very shorter the membranous, and the longest the penile urethra, and come out with the urethral meters. This is the in-depth knowledge about the, and what is the intratesticular duct system, and you can get MCQ, and only this tubular recti, reticuli, and vasa efferentia come under the intratesticular ductal system. Epididymis do not come. These are all the MCQs usually asked. So let us move next. So our main topic, that is the glands, accessory glands. So we have to deal in depth what are their secretions, and are the pH of the secretions, and the constituents of the secretions, and how much percentage of these secretions constitute the semen. So first to name, one is the, as we all know, seminal vesical gland. Another is the prostate gland, and last to name is the Cowpers gland. So first let us take the seminal vesicle. So the basic concept behind these glands is that all these glands secrete some fluids, and all these fluids constitute totally combined constitute the semen. So every one constitute some percentage, and it differs from one gland to another. And with this, let us pursue. So seminal fluid is a paired type of gland, and it is a simple tubular gland. It is a simple tubular type of a gland, and its secretion is alkaline, viscous, and nutritive. Why nutritive? Because presence of fructose. That is very important, because fructose is acting as a helps in the acts as a nutritive to the spermatozoa. And next, what are what are the constituents? Constituents are generally fructose, to name fructose, and potassium citrate ions, and inositol, and prostaglandins, and inorganic phosphate, and other proteins. All these are the constituents of the this fluid of the seminal vesicle. So at last we will be going to the what's the percentage. And next, let us come now I will be discussing one important point about the fructose, and also the importance of prostaglandins. So to name, fructose is not produced anywhere else in the body except the seminal vesicle, either in female or male. There is no production of fructose except in the seminal vesicle. So it is not produced anywhere else in the body. So but when fructose is found, presence of fructose in the female genital tract. It confirms the sexual intercourse. How? Because as if the females in the female genital tract there is no fructose usually, but and the fructose is present in the is formed in the seminal vesicle. So if the fructose is found in the female reproductive tract, it means that there is a transfer of some seminal fluid from the male to the female. That means there has definitely occurred a sexual intercourse. So this helps in the forensic test for the rape victims. So this is very important about fructose, that it is not produced anywhere else. You have to remember this point, only then you can relate it with this forensic test for the rape victims. So that is a very important point. Next, let us move and study the importance of the prostate gland. Is one is the most important is that it helps in the movement of the sperm, and another it prepares the mucus of cervix to be more receptive for sperm. So it prepares the mucus of the cervix of the female to be more receptive, and also helps the movement of the sperm. These are all the important things about the seminal vesicle and their constituent. And next, let us move towards the important prostate gland. So prostate gland, first we will be naming that it is paid or unpaid. It is single gland, it is not paid, and the fluid is generally it is slightly acidic. Fluid is slightly acidic. And what are the constituents? Constituents are usually proteolytic enzymes. Calcium is also present, and citrate ions and phosphate ions are also present. And all this helps in the, and this you have to remember, it helps in the activation of the spermatozoon. So this helps in the activation of, this is the important thing. Now now let us study the how much percentage of this secretion in the semen. So it constitutes, the prostatic gland constitutes nearly 15 to 30 percentage of semen, nearly 30 to 15 to 30% of the semen, whereas the seminal vesicle, the secretion of seminal vesicle constitute nearly 60% of semen. It means there is a larger proportion of the semen coming from the seminal vesicle. So now let us move to the Cowpers gland. So Cowpers gland is also known as the bulbourethral gland. So next, this Cowper gland is paid type of a gland like the seminal vesicle, and the secretion is slightly alkaline. It is very important because next I will explain its importance. And it also has mucus. Please remember, it is alkaline. Why? Because it relates to the function. Very important, you can relate it to the functions of the this fluid, because this Cowpers gland releases its fluid just before the ejaculation, and its function are very important. You can relate it to the alkaline, the pH of the fluid. It helps in the neutralizing acid of urine, So acids and it also lubricates the penis. So this alkaline nature. So in now you will understand that why I have said this is important, because it, as when you remember the function of these secretion of the Cowpers gland, that is a neutralization of the acids present, acids of urine or anything else, as any other acid. So if in order to neutralize this acid, this should be in the pH of the fluid should be the alkaline. That is what how you have to relate the things and study so that you can learn uh, better. So, and it also helps in the lubrication of penis, and as it helps in the neutralization process, it is known as a flushing agent, also known as a flushing agent. So at last, the percentage secretion of this gland constitute nearly negligible. There is no percent, nearly negligible, it is not counted. So with this, let us move forward. So after all these insemination process and all those things, the question arises that what actually is the sperm constitutes of? What are the we know that sperm is present in the semen, but what are the other things that are present in this semen, and what is the pH of this semen? So to know all those things, let us move out. The semen, the pH of semen is slightly alkaline, that is 7.5, whereas, and this semen can be divided into two, that is a seminal plasma, like the blood plasma, seminal plasma, and the sperm. These are the constituents of this semen. And to go in detail, so what are the percentages of this semen? So let us move forward. And first let us take the fluid and sperm that are coming from the vasa difference. This constitute nearly 10% of the semen. And second, let us take the fluid from the seminal vesicle, and this nearly constitutes 60%, as we already know. And the third, let us take the prostate gland, and this nearly constitute the 30%. And we know from the Cowpers or the bulbo-urethral gland, it is nearly negligible. So all these nearly constitute the 100%. So when we go into the analysis of these things, we can analyze that the maximum percentage is coming from the seminal fluid. That can be asked as an MCQ. At last, before ending this class, I would like to add some few extra points to this. That one extra point we have already known, that is the per ml of the semen. If you count the number of sperms, it is nearly 15 million to 200 million sperm count per ml of the sperm in a natural, normally healthy male. So, the second important thing, what is the lifespan? What is the lifespan of sperm after ejaculation, not before, after ejaculation? And what is the lifespan of ovum is also the question. So so the answer to this question is that it is nearly 24 to 48 hours, and ovum lifespan is nearly 15 to 17 days, and it is hours. You have to remember all those things in depth. And but there is a way that we can store these sperms and increase the lifespan of these sperms to nearly days to years as well. And this technique is called cryo preservation. It is being used nowadays, in which the sperms are stored at -100 degrees Celsius. -100 degrees Celsius. That is how you have to relate all your knowledge with the present techniques, present new technology, as well as we have to interrelate the concept. So with this, we end our class today, the part 1 of the male reproductive system under the human reproduction. And further, we will be dealing with the separate class with the spermatogenesis under the gametogenesis and so on. And at last, I thank you very much for watching me. Please, if you like, please like, share. Thank you.